Hi, welcome everybody. We're going to give people a few more moments to sign on and then we'll get started. You are welcome to chat amongst yourselves using the chat. All right, I think we should go ahead and get started. So hello, welcome everybody. My name is Erica Reinfeld. I manage the public galleries at the Koch Institute at MIT. You can see them behind me, although I'm secretly calling in from home. And I am delighted to introduce part 2 of our showcase of the 2020 Image Awards exhibition, 2020 Visions. We've got 5 images for you today. And I want to welcome those of you who were here for us on Tuesday. We heard 5 images on Tuesday and we've got another 5 today. The way this is going to work is I'm going to introduce the presenters one at a time. They're going to share their stories through some slides, talk about the images and what went into them. And then after each presentation, we'll have some Q&A. So as they're presenting, there's a Q&A button at the bottom of your screen. Use that to type in any questions you have for them. I'll try to get to as many of them as we can. You're also welcome to use the chat for informal conversation and comments. If it's not a formal question, but you want to share something, please go ahead and use the chat, talk amongst yourselves, and then we will do that for all 5 images. I have to let you know that we're recording this. We'll be posting this on the website along with Tuesday's talks. So if you missed that, definitely check those out. We heard some great stories and I am excited to share this next batch of images with you. We're actually going to start off with the image at the far side of the gallery over there. If you could enter the Koch Institute building from Vassar Street, this is what you'd see. It's the first scientific image story, but unlike all of the other images in the galleries, it was not taken by a scientist. Keith Ellenbogen is a professional photographer and a visiting artist at MIT Sea Grant. And he is actually out in the field right now. I think he's the furthest afield from the Koch Institute building, but has graciously signed on to give us a behind the scenes view of this image and how he uses photography as a communication tool to tell visual stories about the marine environment.
Hey, Erica. Thank you very much for the kind introduction and words, and hi all. So I'm an underwater wildlife photographer and am currently working on a National Marine Sanctuary Foundation Hollings Ocean Awareness Award to use the art of photography and microscopy to connect people to the extraordinary underwater world within Stellwagen Bank National Marine Sanctuary, from ocean giants like humpback whales and great white sharks to the microscopic photosynthetic dinoflagellates like these three serratium that in their vast numbers generate much of the oxygen we breathe and form the basis of the food chain. To collect the samples, we set sail on board MIT Sea Grant's boat and traveled to Stellwagen Bank National Marine Sanctuary, just 20 nautical miles off the coast of Boston. And as part of this project, we are engaging students in all aspects of plankton collection and image-making techniques. This gives students at MIT a unique hands-on experience in how art and science can enhance visual communication and storytelling. Our process is to conduct a plankton tow, collect a concentrated sample, and bring the living plankton back to the lab, where I put them under a microscope and apply my skills as a wildlife photographer, focusing on light, composition, color, rhythm, and movement, all to create an emotional connection to these microscopic creatures. Each slide is an opportunity to explore and discover. This exotic-looking creature is a radiolarian. Dinoflagellates are a large, diverse group of single-celled algae that are of immense importance to the marine environment. And in addition to photography, video can reveal the rapidly undulating flagella and the current that it creates. And just to the left is algae. As part of my image-making process, I like to use a combination of dark field, polarization, and image stacking techniques to illuminate the subject to showcase a full focal range. The blurred tips of these dinoflagellates reveal a subtle movement that elicits a sense of motion in the overall composition. Even within the microscopic world, there are signs of pollution. Unfortunately, this image shows two dinoflagellates intertwined with microplastics or microfibers. Additionally, I've been experimenting with other photomicroscopy techniques. For example, the red fluorescence within this image shows a distribution of chlorophyll within the cells, as well as a visual expression of ruby red. Or using a scanning electron microscope to create an extreme close-up view that reveals intimate insights into the complex body, shape, and texture. But the image that I'm most proud of is this composition of three interconnected serratium that are currently on exhibit at the Koch Institute Public Galleries, in which their microscopic size is exhibited exponentially larger than life, symbolizing the gigantic impact dinoflagellates have on our planet. The project is a collaboration that involves many people and organizations, and I'm eternally grateful for the support and encouragement. And finally, I just would like to thank the Koch Institute of Public Galleries for this opportunity. I hope you're all able to see this exhibit during these COVID times. Thank you so much, and if you have any questions, I look forward to answering them.
Thank you so much, Keith. So as he said, if you've got questions, please share them. I think what I'd like to do while we wait for people to warm up their typing fingers is just ask you about some of the challenges of collecting these things. It's obvious once you've got the sample, there's a lot that you can do to explore it and tell stories with it. But what about the challenges that go into just getting that?
That's such a great question. You know, it's so interesting. The way this works is you do a plankton tow for several minutes and you collect a whole sample and then you bring them back to the lab. But even within this large concentration that's really fairly small, maybe the size of a large bowl, is a whole world of discovery. And just finding a couple of little drops in there, you know, doesn't guarantee what you're going to find. So I don't know how many millions of drops make up a bowl of water, but that number is what you're looking at. And so you hope within that slide that you find the right creature that you're looking to photograph. Oh, hello?
Oh, sorry, I think we've jumped into a different SEM image that's coming up next. So we have a comment actually saying that they love the electron microscopy shot even more. So you're in luck because we got a lot of electron microscopy coming up later. Yeah. So Keith, was there more you wanted to say about the collection process?
Well, I think no, I think that was pretty good. But the thing that's most interesting is that even with a sample that you have, you still have to hunt and peck within that concentration to find things. And you spend a lot of time really looking around. I think people don't realize how much time you spend looking for these microscopic creatures within the within a bowl and then ultimately on the slide.
Yeah. So you and I actually spoke back in June at a program that the MIT Museum put together about imaging science and science photography. And one of the things we talked about there was this idea of all this preparation that has to go into creating catching a spontaneous moment.
Yeah. Yeah, you know, it's so interesting. As a wildlife photographer, you really look for the animal's body gesture, its language, and how to create an image that people can connect to. And I spend a lot of time not just looking for the creature, but looking for a way that it visually is engaging, connecting, or a composition that I like, the negative space, the relationship between two creatures together. And so from an art and photography point of view, you're always looking for that. And then also from a scientific point of view, what's nice is sometimes you're just looking to document what it is, but in storytelling, you want to create an image that has multiple layers. And so when there's a few extra things that can connect one to the next, it really becomes something more dynamic. And I think as a photographer, what I really spend time doing looking through craft is finding those moments where you can position things. And the slide is so wonderful because you can make these micro adjustments, moving it and moving the composition. And so you can really create what you sort of begin to see and envision if you get fortunate enough to find it.
Yeah, I was going to ask you to comment on the interplay between you as the photographer and the researchers that you work with, because you're working very closely with researchers.
Yeah, it's really a wonderful experience to work so closely with researchers because I get the luxury, and I'm spoiled because of the people that I'm working with, but I get the luxury of learning from them. So they'll say that this is really particularly important, or to focus on this, or to think about that. And so it just gives me a driving factor in terms of my image making, what I'm sort of trying hard to photograph. Maybe it's the color, maybe it's the shape, maybe it's a little movement that I might get lucky, like the flagella moving, or who knows what, but that sort of relationship allows me to be much more honed in and much more focused on the elements that actually help exemplify what makes each creature so unique.
That's excellent. So we have an anonymous attendee who's wondering about where you are, and I know you're on your mission here, but maybe you could just tell us a little bit about what's coming next for you.
Yeah.
In this
so
project.
I mean, for me, the work that I focus on is being in Stellwagenbank Sanctuary, which is really off the coast of Boston, just 20 nautical miles from Boston, and right in between the North Shore and the South Shore. So I spend most of my time now on the South Shore of Boston and trying to get out to sea as much as possible during the weather. And so, you know, the fortunate part for me is that, you know, what's so interesting about the plankton creatures is you have to bring that back to the lab, but so much of the wildlife is out in the field. And so this is the joy of this is that I get to go into the field to go capture the animals and then back into the lab to photograph them.
Excellent. All right. Well, we could talk for hours about any one of these photographs, but I will encourage people to keep feeding questions and comments into the chat, and we're going to actually move on to our next presenter. So Rameen is going to share his electron microscopy image here. So this next image is a different kind of underwater journey. It's called 10,000 Nanoleagues under the SEM. It's kind of a visual wordplay there. And it's going to take us on a different undersea journey. Before Rameen begins presenting, I do want to give a shout out to Paramesh Karandikar, who was the technician who originally took this image and submitted it along with a bunch of others this fall. He started med school over the summer, and apparently that's keeping him fairly busy. So I'm very grateful that Rameen was willing to step in as the project lead in the Langer Lab and present this image in Param's stead.
Thanks, Erica. Can you see this?
Yep, looks great.
Okay, so like Erica, like you mentioned, I'm just basically filling in for Paramesh, who was the research tech that was in this project. I want to explain to everyone how we came about this very insightful picture and the research that goes behind this. I want to start off about just explaining how we came about what we're doing. My background is as a cardiologist, an academic cardiologist. My medical background is on electrophysiology as sort of a day job where we're putting pacemakers, defibrillators in. And my PhD was on looking at materials, in particular materials related to energy harvesting. And I worked on stem cells in particular, induced pluripotent stem cells, which are stem cells from us as humans and which can be differentiated to become other types of cells. And in my case, heart cells. And I'm here at the Lange Lab at the Koch, which is a tremendous environment where we have a lot of interdisciplinary work and a lot of interdisciplinary work that actually has impact in the clinic. And we're sort of playing around with a lot of the sort of material science, polymer science, and implications of that. And one of the points that got my interest was, how do we look at harvesting of energy in a safe, sustainable, and efficient way? And a lot has been done in this field, but very much in the way of research, but very few in the way of accessibility. And how can we really push that boundary for medical use, and in particular clinical use in general? And one of the points that I was interested in was sort of this concept of batteries. So we're all afraid what batteries do, and we know what batteries are accessible. But you know, with our current point and with the environmental issues related with batteries, we wanted a safe and renewable source of batteries. And one of the points that I was keen to involve was the application of cellular components. And what I meant by that is, given our interest and given my background in stem cells, can we use stem cells, and can we use our sort of kinetic power in beating heart muscle, heart cells, to initiate that process of external battery use and harvesting that. And we sort of played around technically, and very happy to announce that we've sort of moved on from how we extract these cells. We have a very unique system of doing that. We extract cells from urine, normal urine, and we engineer those cells to become the very stem cells, the induced pluripotent stem cells, and which are then differentiated. That means we are putting chemically defined components to shift the whole reprogramming of that cell, of that stem cell, to become a beating heart cell. And we're talking about doing that in a larger scale, and that's what we've sort of been working on, and we've sort of optimized that, so we're getting lots and lots of these beating heart cells, and you can see that as a whole raft of beating cells, similar to what happens in the heart. And then so that movement, that sort of natural kinetic movement of beating heart cells lends itself to a very interesting source of power, of kinetic power. And the key differential here was, how do we take that kinetic power, which is really just similar to what you and I are doing now when our hearts are beating? How do we try and see, can we access some of that? Can we even harvest some of that energy? And can we harvest that energy to an order of magnitude above what's currently possible? And we were looking at different materials. One of the materials that we are particularly interested in is zinc oxide. And zinc oxide has been used previously because it's a very conductive, cheap material to use, but it's one of its key characteristics is that it has this ability of being able to develop a charge when you push on it, when you mechanically deform it. And that's called piezoelectric characteristics. And what that allows the zinc oxide to do is take that sort of change in deflection or of movement, and take that kinetic mechanical energy and convert that to the energy that we want to do, which is essentially to store that for battery use. And our thought was, can we produce this zinc oxide, but at a much, much smaller scale, in particular in the nanoscale? And to give you a context of that, we're talking nanoscale is 1/1000th of a meter. So we're in the sort of ranges of below and sort of in that realm of DNA. And we wanted to go to that lower levels because you say, if you have at such a small scale, if you had almost little minute nanoscale farms of this, can we sort of use that as a means to upscale the whole process of harvesting? And so it led us down this track of active research that we're still working on. And so this is how this was all born about, that we wanted to understand the properties and behaviors of these zinc oxide. And if we were to miniaturize it, what are the implications of that? And from my particular sentiment, I was very interested because obviously this has implications for biosensors. The way we currently put defibrillators, pacemakers always require an external source of energy. Could this be something to look at? And what we really wanted to do is, how do we know that we're producing the zinc oxide? This is obviously a chemical process that we were developing. And this is where Paramesh was a real project that I was sort of guiding him towards, to say, look, we're now trying to make homogeneous zinc oxide, and we want it to be as a layer. So can we get these almost hairs of zinc oxide to see if we can deflect that? And so to characterize that, because it's in that nanoscale, we used the sort of SEM images as you'll see, and it's a very easy way, but also a definable and objective way to characterize the properties that we need. So it was a very easy way to say, yes, has the chemistry worked, or no, it hasn't. And then I wanted to share with you where this whole stem of the discussion came from. Why did we pick the term as it is? And I just want to spend a bit of time on that. It's an interesting story. So Jules Verne, a French author, was writing a lot on science fiction. And one of the major books back in 1869 was 20,000 Leagues Under the Seas. And the French title you have there, De 20,000 Lieues sous la mer, Tout le monde sous-marine, is really the world tour of underwater. And what he described is very interesting, of this submarine-like machine called the Nautilus, which was discovered by people who had left New York, and they thought they were seeing a sea monster, and it actually turned out to be this Nautilus. And what the Nautilus was doing, it was going around the Pacific Ocean and seeing different oceans and traveling across the floor bed of the oceans, and it would created a whole new world for people to view. Now, the book, obviously, 100 years before the whole instigation of submarines, was a very insightful view of where we are today. But there's a very interesting point which we wanted to capture in that whole book, which I think says a lot about why we chose that title. And that is, the whole option there is, the book 20,000 Leagues Under the Seas talks about where humanity and sort of the whole notion of trying to live for a better humanity using science, using sort of less pollutable forms of understanding, and using science to actually see. And that was a very interesting point that came out from this discussion. So when we were looking at the zinc oxide, that's one of the things that really comes out, that we are using science at this nanoscale, and getting information was something that just really stood out from us. And then when we first started, I'll draw your attention to this picture on the left of orange. This is what we initially got. And these are what I would call the weeds of this garden of zinc oxide that we're trying to get. And yes, they look pretty, but it's not actually what I wanted. I was, when we first did this, and Paramesh showed me this, I was like, okay, but that's not what we want. What we really want is the diagram on and the picture on the right, where you see in green these sort of spikes of zinc oxide. These are sort of whole trails that we wanted to see, and you see it within this jungle. The perspective there is there's a jungle of zinc oxide which are in sort of this structures, towers in the nanoscale. And we, in the intermediary of trying to aim for that homogeneous garden, this picture that was selected by the judging panel came up. And again, we were sort of transitioning from different types of chemical processes to get to the end of this homogeneous garden, and then coming from total weeds or these sort of little plants of the zinc oxide which hadn't formed properly, we got to an intermediary. And that sort of came out that when you look at this picture, you focus on the fact that actually, oh, it's the center point that we were trying to actually discover or actually trying to find. But that's not the case. The case actually is, from a scientific perspective, it's the bits around it that we were trying to get to. And this was a distraction, but it shows, and what really stood out is that it's sometimes in science, to get to the end goal, we have to sort of take stock of why things are not happening appropriately and optimize. And this is a good example of that we're not fully there yet. And this picture was taken at that point where we hadn't optimized the whole process till we had found our optimum sort of set of conditions, which you saw in the previous diagram. And so that made us really, it came out to me that this was a very important point to make. And Paramesh really liked the sort of the title of it. And it sort of rang true that when you look at things at this scale and you see this, you know, the science that we're doing is very intricate, and sort of every little option of that comes through. And so that was one of our defining points. And the beauty of this sort of snowflake-like item in the middle is to sort of really show that sometimes when we're looking at things, the focal point may not be the major output that we're seeking. And so this is the goal, as I said, so trying to make more of these lawns of zinc oxides in a homogeneous way. We have done some testing and we're already getting some power output. And there's still a bit more to go to actually optimize that whole sort of efficiency, but it's really directed us attention in the way that we want to move for our end goals. So I want to thank everyone, especially Paramesh, who obviously he started med school, and so he couldn't be here. And David Mankus, Margaret, Abigail, who are all part of the sort of the Peterson Nanotech Materials core facility, and they were integral in the sort of taking the picture and sort of characterizing this whole process for us. So we're eternally grateful. Gio Traverso and Bob, obviously here at the Koch, as our mentors, and obviously everyone in the Koch Institute, which as an institute still continues to inspire and is a wonderful environment where we can sort of share and have this sort of interdisciplinary discussion. So I hope that's, that was, I want to end here.
Thank you so much, Rameen. I think Paramesh would be very proud of the way you've represented the images. We do have a couple questions in the Q&A.
Sure.
Oh, start my video there. Yes. A couple questions in the Q&A, and in the interest of moving along, I'm going to encourage maybe to answer some of the more technical questions over there in writing. But Tina was asking about the difference between the different parts of the image. So I guess I'm wondering, can you comment on how you take an image and you look at something and make a decision about what you want to do next in the lab based on seeing the image?
Yeah, so so the main point is we were looking at these sort of homogeneous layers there. And you saw we had the spikes. We had artificially colored them just to sort of for our own understanding. And so it sort of made it easier to have sort of very bright colors to say if it's not homogeneous or it's not straight, we know exactly where those areas are. How do we do that? That's a great question. So initially, as the chemistry developed and the process develops, we take individual segments and they're done repeatedly across the entire segment. And so the figures that you see are a representation of, say, 90% of the entire surface area. So that's how we sort of looked at it. But you're right, it's a bit of a laborious task to saying, are you looking at every single corner bit because it's in such a small scale? But what we had done is once we had done the initial sort of chemistry and we had come to that fruition, we were able to sort of upscale and then go back and sort of check in a random fashion to say, how often are we getting those sort of spikes?
Excellent. Thank you so much. And I'll let people know that David Menkes, who's one of the microscopists behind this image, is also on the call. And so he can potentially answer some questions as well if you want to put them into the Q&A or the chat. But what I want to do now is move on to another image that is behind me. This is visualizing vasculature, and it actually looks very much like some of these nature analogies that we've been seeing, but in fact, this is the image that is most closely aligned with the 2020, most literally aligned with the 2020 visions theme. I think all of our image present a vision for the future. This is one that really gets into the fundamental biology of things behind actual literal vision, but it has implications for a lot of other things as well. So Genevieve, why don't you share your image with us? Which we saw one moment ago.
Can you see it now?
No, try, we're gonna reshare. This happened in the dress rehearsal too. There it is, perfect.
Okay. All right, thank you, Erica. So I'm a postdoc in the Heinz lab, and today I just want to briefly describe what my image is to you all. It may look a bit like a flower, but it's actually an image of a retina showing the blood vessels that grow on the surface of the retina. And I'll tell you a little bit about why we're interested in studying this. So the vasculature, or the circulatory system, is a network of blood vessels that carries blood all throughout our body to deliver oxygen and nutrients to all of our tissues. And this is critical for all the tissues in our body to function properly. And importantly, tumors also rely on a network of blood vessels to provide nutrients to support their growth and survival. Unfortunately, however, having this network of blood vessels also provides an outlet for cancer cells to escape the primary tumor and spread to other parts of the body, which is what makes cancer so deadly. So given this important role that tumor vasculature plays in both the growth and metastasis of tumors, it's been quite an attractive target for cancer treatment. However, so far, nearly all of the therapeutic interventions that aim to target the tumor vasculature have largely failed in the clinic. And we don't completely understand why. So what we want to do is gain an even better understanding of how blood vessel networks normally develop in our body and the factors that control this process, which could provide us in the future with new and possibly more effective ways of intervening with the formation of blood vessels in a tumor. So a great place to study the normal formation of blood vessels is the retina, which is a tissue layer at the back of the eye, which collects light and transmits these signals to our brain through the optic nerve. And the retina contains a highly organized vasculature network that provides blood to the retinal neurons. And this is critical for maintaining proper eyesight. And this is a great model to study vasculature development because in mice, these blood vessels actually develop over the first 2 weeks after birth. And so this allows us to more easily visualize the vasculature at various time points over the course of this development. And you can see here in these images, the progression of blood vessel growth from day 1 to day 9 after birth, where the blood vessels, which are shown in fluorescent green, begin growing at the optic nerve at the center of the retina, and then progressively begin to migrate and grow out radially towards the edge of the retina, until by day 9, they've reached the edge and cover the entire surface of the retina. And so this system really gives us a great opportunity to more easily identify genes or other factors that may either promote or perturb the growth or organization of this vessel. So vessels grow by a process called angiogenesis, and this is when blood vessels begin to sprout and generate new vessels from the pre-existing ones. And the migration and survival of these cells that make up the walls of blood vessels depend on interactions with their surrounding environment. And what I'm showing you here in this closer view of the retina on the right is that the vasculature, which is shown in this yellowish green color, develops on top of an underlying meshwork of protein fibers known as the extracellular matrix, which is shown in red. And in this image, if you look carefully enough, you may be able to see that the cells at the tips of these sprouting vessels actually reach out and directly adhere to these underlying protein fibers. And so in this way, the cells of these sprouting, growing vessels can receive signals from this underlying meshwork of protein fibers. And this helps them to both survive, but also helps guide the direction of their migration, so that they follow these pathways that have been laid out beneath them all the way out to the edge of the retina. And so what we're doing in the lab is we're studying these interactions and how this underlying network of matrix fibers regulates and maintains the proper growth and organization of this vessel network. And we're hoping to identify new proteins that may exist in this matrix of protein fibers that might participate in this process. And again, I'll just reiterate that we hope that in the future, studying the normal formation of blood vessels might offer clues as to what stirs the formation of blood vessels in a tumor, and this may offer a new avenue for therapeutic intervention in cancer. And so with that, I would like to thank my advisor, Dr. Richard Hines, and the entire Hines Laboratory, and I'd like to especially thank Tao Nguyen, who initiated this project during her PhD studies, as well as Jeffrey Kuhn from the Koch Institute Microscopy Facility, who spent quite a while helping me to figure out the best way to image these retinas. And thank you all for listening, and I'm happy to take any questions.
Thank you, Genevieve. Can you say a little bit about how you made this image? We kind of get a sense of the biology that went into building this structure, but how do you actually put together this image? I think it's deceptively simple looking.
Yes. Well, it was very challenging because the retina was larger than what could fit in the field of view of the microscope, or the image size that the microscope could capture. So Eric and I really spent, Jeffrey and I really spent quite a bit of time trying to figure out the best way to image the entire retina. And what we decided to do was to do paneling, where we took, this is actually a compilation of 16 different images. So we take 16 smaller images of the whole retina and then stitch them all together and do some fancy programming to get them to stitch up perfectly so all the vessels align. And that was quite challenging. I also want to say that another challenge of this is that the retina is a spherical cup shape, and what you see in the image is flat. And so that is why it looks like a flower, because we take this cup shape and cut some slices into it so that it essentially has petals that we can fold down and flat mount it on a slide and then be able to image it in the microscope. But it still isn't totally flat, and so that leads to a lot of focus issues, and took quite a while for us to figure out the best and most efficient way to image these. But in the end, we came up with a pretty good and efficient method, and so that's what you see.
The effect is great. Yeah, there are a lot of layers to it, I guess.
Yes, yeah.
Quite literally and figuratively. So you mentioned the blood vessels feeding tumors. So, right, we're at the Koch Institute. Let's talk a little bit about the cancer here, right? The blood vessels feeding the tumors, and your goal with the project was to look at the interplay between the blood vessels and the proteins. So what role do you anticipate the proteins having in kind of controlling cancer or affecting how cancer works?
Yeah, so I think that the proteins that are there in the tumor are really just creating this supportive environment that allows the tumor to co-opt surrounding vessels that are in the normal tissues and allow the angiogenesis to happen. And so these normal vessels can then sprout throughout the tumor and start growing. So the proteins in the tumor really allow the tumor to hijack everything else that's going on in the body and bring in the support and nutrients it needs into the tumor. So, you know, if we could figure out what are the important proteins there that are doing that, we could maybe target them and block them so that the tumor isn't able to really harness that ability to take over the vasculature and grow new blood vessels. You know, if you can restrict blood vessels in the tumor, then in theory, the tumor can't have an adequate supply of oxygen and nutrients and would die or shrink, or wouldn't have, yeah, I guess, wouldn't have access to appropriate nutrients, or access for the cancer cells to escape and metastasize to other organs.
That makes a lot of sense. Sounds pretty important. So thank you so much. Keep up the good work, and I hope it continues to go well. All right. So Genevieve just alluded to cancer's very nefarious, sneaky ways, and that's actually a great transition into our next image. We've got some natural killer cells coming up, which, like any immune system, tends to be a story between good and evil. And this particular story for Cloak and Swagger, presented by Arnav Chhabra, is going to kind of turn that good versus evil, sneaky cancer cells on its head. So Arnav, if you would like to, there you are.
Can you hear me?
I can hear you.
So let me just get my screen shared. All right, how does that look?
Oh, looks good.
Awesome. So thank you for the kind introduction, Erica. My name is Arnav Chhabra, and I'm a postdoctoral fellow in Dr. Sangeeta Bhatia's lab at MIT. So at the Bhatia lab, we are a team of engineers who like to derive inspiration from biology. So as an example, we'll take a novel scientific finding and leverage that discovery to develop a tool that will either help us with early detection of disease or help us improve an actual therapy. So for this project, the scientific principle that underlies our work is also the basis for the burgeoning field of immune regulation. In 2018, the Nobel Prize for Medicine was awarded to two scientists, Drs. Jim Allison and Dr. Tasuku Honjo, for discovery of a new mechanism that allows cancer cells to shield themselves from the immune system. This phenomena is in fact leveraged by a new class of drugs called checkpoint inhibitors that many of you may have heard of. As you can see, cancer cells overexpress these checkpoint molecules on their surface, and really what these molecules do is they serve as stop signals to dampen the immune system. While this phenomena is largely used by cancers to harm the body, we wondered whether we could learn from the enemy and repurpose similar biology for the benefit of human health. We looked at many different applications, but the one that really spoke to us was the field of organ transplantation. For people with end-stage organ failure, their only option is a transplant. However, the supply of these organs is very limited. As you can see, the organ waitlist has rapidly increased over the last 20 years, while the number of transplants and donors has not kept up. While there are many reasons for this gap, a primary one is that transplants require immune matching so as not to trigger a severe immune reaction. This matching requirement severely limits the number of people who can receive a transplant. So we thought to ourselves, what if we could utilize the same biology that cancer cells do, but instead to make universally compatible organs that don't require matching? This could really help us increase the supply of donor organs. And that's what we did. We engineered a molecular cloak that allows transplant cells to dampen the immune system and thus protects mismatched grafts from rejection. As an example, if you have a mismatched graft without our molecular cloak, immune cells of the recipient will recognize it as foreign and destroy it. On the other hand, if you activate a molecular cloak using the genetic circuits we have engineered inside of these cells, it creates a virtual shield around the graft that helps protect it from immune cells. In order to test our invention, we took the engineered graft cells, activated the molecular cloak, and put mismatched human immune cells on them. By using scanning electron microscopy, we were able to visualize what was happening at the cellular level. Whereas in the control setting, the grafts would get destroyed by immune cells, with the activated molecular cloaks, here's what we saw. We saw that graft cells, shown here in purple, were able to coexist with the green immune cells without any real damage. immune cells typically use these projections called microvilli as sensors of foreign material. But in our experiment, while they were able to recognize the graft as foreign, they were not able to actually kill it. In essence, our molecular cloak was functioning how we wanted it to. And with that, I'd like to acknowledge a whole host of people who have helped make this project possible. I'd like to thank our entire lab, specifically my PhD advisor and longtime mentor, Dr. Sangeeta Bhatia. I'd like to thank the Robert Swanson core facilities at MIT, and David, Margaret, and Abigail from the Peterson Nanotechnology Materials Facility for helping with all of the electron microscopy work that I showed. Lastly, I'd also like to thank Erica for running the Koch Image Awards and the 2020 judges for selecting our work. And with that, I'll take any questions.
Thank you, Arnav. Well, we've already got a question in the Q&A. And I think maybe leave your slides up for this because it's a question about the fronds that are coming off the immune cells. One of them looks broken.
Yeah, so I think that's a great question. And that was one of the questions we've received many times whenever I've showed this image. And it's an important one because I think with electron microscopy, you often have what we call artifacts. And I may actually let David, who's also on the line, answer the question about artifacts. But these typical things that are coming off of the immune cells, they're called microvilli. And these are projections. It's actually still kind of unknown what the projections are for, but it's been hypothesized that they're either sensors or transducers. So in many cases, they can sense foreign material. And in many cases, they can transduce the killing of foreign material. So the cytotoxicity that is often associated with the T cells. And the breaks that you're seeing in here often have to do with the processing of the material. So I'll let sort of David answer that question.
Sure, I can speak to that. Let's see, okay, there we are, sorry. I think the video might be up
Hi,
now.
David.
Hi. So I can speak to that. The processing that we do for electron microscopy requires us to actually dry out the cells completely. So instead of being very hydrated and sitting in their happy medium that they like to live in, we actually fix them with a variety of different chemicals that makes their shapes very stable. And then we go through a long processing that actually replaces all the water with organic solvents, and then we dry them out completely before entering them into the microscope. While this does preserve the vast majority of the morphology of these cells we're looking at, some small artifacts like a little bit of shrinkage or a little bit movement of the different parts of the cell might occur. And so in this case, that frond that is attached quite firmly down on the cover slip that these cells are living on is remaining in place because it's attached firmly, but the little microvilli that's reaching out there shrinks back a little bit toward the cell, and so it breaks and there's a small gap there. And we can see that repeated in several of the other microvilli around these cells. The actual gaps there, however, are only a few nanometers and don't represent a large change in the way that these cells actually look or behave.
Thank you so much, David. Yeah, scale is always a challenge, especially when we then blow these things up to be 8 feet tall in our light boxes. Arnav, could you talk a little bit about, you say the electron microscopy is really important for observing what's happening with the immune cells. So how does what you're observing with the immune cells change what you do to the graft cells? Because it's the graft cells that you're actually engineering.
Right. So I think we care a lot about the interaction of the graft cells with the immune system. And the observation is kind of important for us because we actually want to know what's happening at the cellular level. You know, oftentimes we'll do these animal experiments that'll give us a sense of whether the graft can stay alive in the mismatched immune setting. But what we often don't understand is how that actually looks on a cellular level. And the cellular level, in many cases, is what kind of dictates how we engineer the graft. Because if we see that the cells are facing an immune attack but not dying, that is a different phenomenon than whether they're actually creating a shield that even prevents the cells from getting to them. So a lot of people have looked at sort of creating these cloaks, and these are like physical barriers, right, that prevent the immune cells from even getting to the graft. Our strategy is a little different in that we don't have a physical barrier. What I like to call is that we have a biochemical barrier, which is the cells will come find it, the immune cells will come find it, but they won't necessarily know what to do once they're there, if that makes sense.
It does. You couldn't hear me say it. It makes sense. And I really appreciate you sharing that with us. I think we're going to move on to our final image of the afternoon. Like I said, those of you out there, if you've got questions, feel free to pop them into the Q&A or use the chat for other conversation. So our final image is we're going to take a little deep dive into some cancer biology and tumor initiation. We shall end with the beginning. Another case of looks like a cancer cell, acts like a cancer cell. What's going on here? So George Eng, who is speaking to us directly from the Yilmaz lab at the Koch Institute, is going to present Co-Culture Club and give us a very well-rounded view of tumor initiation.
Hopefully, yeah, that's great. Thanks for the introduction, Erica. And yes, my name is George Eng. I'm in the Yilmaz lab, and I'm going to present the work I did with Jonathan Braverman, another postdoc in the lab, to generate the image.
George, can you switch to presentation view rather than presenter view?
Oh, sure. Yeah, sounds good. Is that better?
I'm still seeing, there we go. Perfect. Thank you.
Okay, yeah, sure. Right. So, right. And the title of this is Using Organoids to Model Intestinal Regeneration and Colon Cancer. And so by way of background, one past one, this is a histologic image of the normal colon. And, just like Omer, I'm also a pathologist, which is a doctor who, and one aspect of our job is to look at histologic slides. And it involves the visual diagnosis of disease by taking little bits of tissue and looking at it under a microscope. And so the normal colon is part of our digestive system. It has a lot of interesting and important functions. And one of them is to resorb water from our digested food. That's, critical for our survival. And one thing that's unique about it is that it's arranged in these units called crypts, which are test tube-like shaped. And you can see that on the left here in this Alcian blue periodic acid shift stain section. And you can see they have a linear arrangement with the stem cells on the bottom and the differentiated cells on the top. And that's how the tissue regenerates itself. Unfortunately, though, this tissue can give rise to cancer, specifically colon cancer. And colon cancer is still the second most common cause of cancer deaths in the United States, with an expected 53,000 even in 2020. And so colon cancer is actually something we can model in the lab. So here is a histologic image of a colonic tumor. And you can see on the left, there is the normal crypts that we just talked about, where they have the normal organization and the cells are respecting each other's borders, and they are undergoing the same normal process of stem cell renewal and differentiation. However, on the right, you can see that evolving into a tumor. So a tumor has arisen inside the normal tissue and is giving rise to this mass, which is composed of colon cancer cells. And we know for colon cancer, the most common cause is actually loss of function of a protein called adenomatous polyposis coli, or APC for short. And it's the most commonly mutated gene in colon cancer. And we can mimic colon cancer in mice in the laboratory, which we do in the Yilmaz lab, by deleting the APC gene, and we can specifically induce tumors where we want to. And for example, here you can see on the right, we specifically deleted the APC gene in a few of the stem cells, and it generated this tumor in the mouse. One very convenient aspect of the intestine is that we are able to dissociate the crypts and grow them outside the body of the animal or of a person as mini tissues or organoids. And so on the left, you can see an image of a recently or freshly isolated set of crypts that were liberated from the underlying tissue. And then what we do is we actually have to embed those crypts into a hydrogel, which is a jello-like material. It's very soft and friendly for the crypts. And they're able to grow in media that we maintain in the lab. So we put them into these well plates, as you can see on the right, in a droplet format for convenience, which you can see there. And what we eventually then do after the hydrogel or jello-like material has set is we place media on top of it to permit their growth. And so that turns into these images that you can see on the left and the right. So the crypts actually end up growing and ballooning up because they no longer remember which side they were because they're embedded into a hydrogel. So on the left is the normal colon crypts after they've been grown for a few days. On the right are the tumor crypts after they've been isolated and grown for the same amount of time. So on the left, you can see how the normal crypts actually have a very balloon-like and cyst-like architecture and shape. And on the right, you can see that the tumor ones, though they also have a cyst-like and spherical appearance, they have some misshapen and unusual features and sizes. And also, they actually grow at an increased density compared to the normal because the cells are much more clonogenic or have the capacity to renew themselves. One convenient aspect of growing the cells in culture is that we are able to modify them to be fluorescent. Because since they're ex vivo and outside the animal, we are able to genetically engineer them, either using CRISPR or Cas9 or Cree technologies. And so after we make them fluorescent, here's an example of that after we've modified them to be fluorescent. The fluorescent markers allow us to rapidly test conditions which may differentially affect the normal compared to tumor organoids. So the normal cells have been engineered to express a green fluorescent protein, so they look green, and the tumor cells have been engineered to express a red fluorescent protein, so you can see them here in red. So on the left, we grew them in a condition with media that permits the tumor to grow, and actually the normal is not doing very well. However, interestingly, on the right, this media condition selects for the normal cells to grow and outcompete the tumor cells, which is obviously something we're very interested in at the Koch Institute trying to treat cancer. And this is how we actually generated the image that we submitted for the KI award. And so this is that final image that was selected and is on the first floor of the image gallery. And I'd like to acknowledge the people that made this possible, as well as the funding sources. So Omer Yilmaz, the lab PI, Daya Zhang and Camelia Huang, who are the UROPs or undergraduate researchers on the project, the rest of the Yilmaz lab, as well as the JAX lab for their expertise, and of course the KI Image Awards themselves and Erica and her team for putting it all together, as well as the specific core facilities and funding sources that without which this wouldn't have been possible. So with that, I'd like to take any questions.
Thank you so much, George. Excellent. So, oh, here's my video. All right. Yep. So here's the image on that side. So you're a clinician. So there's a lot to learn here about the cancer biology and the initiation of tumors. In the caption, we talked a little bit about how this helps us figure out how to intervene. So as a clinician, what's really exciting about this?
I think one of the most interesting aspects, which is sort of new, that maybe the technology, isn't quite apparent, but that you can grow the tumor and the normal together. So in the past, growing these adult stem cells of the intestine wasn't readily available. And so now we can do that both for human cells and from the mouse. And by doing the comparison like we have in the image of having the two directly against each other, you can screen many different compounds, both drugs, metabolites, or even bacterial species, for microbiome type studies to try to see what better selects against the tumor but promotes the normal. And that's essentially what we try to do with chemotherapy, of course, to try to select against the tumor cells and hope that the normal cells regenerate so that you are able to still have that tissue. But by being able to grow them outside the body and maintain the stem cells, you can directly compare them. And that's something that's only sort of been recently available as a technology. It's only been available in the last, few years. And so the Yilmaz lab is continually trying to explore how to exploit this new methodology to try to identify the best ways of selecting for against the tumor and selecting for the normal.
Yeah, absolutely. I had a meeting with somebody yesterday, and one of the things that came up is where's the field of cancer research heading and what are some of the big innovations? And organoids were they were identified as one of these rising stars. So they have a lot of potential here. Oh, go ahead.
Yeah, one of the interesting things is we could also do it from patients. So one of the things that I do here and as my work at MGH includes, we take samples from patients who have tumors, and we actually grow both the normal and the tumor, and we are able to, for each individual person, identify specific drugs or metabolites that actually cause their stem cells their normal stem cells to grow better and actually inhibit their tumor. And that's something that is very new, and centers are trying to figure out, but it's something we're trying to implement for different patients at MGH, particularly people who are very young, because it's sort of where there's an expectation that there might be something specific that we can treat them with.
Right. No, that's super powerful. And I know in the Yilmaz lab, you're also doing some of these fundamental biology questions, similar to what we talked about with Genevieve, that it's got these very practical applications, and it also has this key discoveries about how the body works, how cells work.
Yeah, it's really cool that you can grow the stem cells outside the body. And so one of the key things that enabled this to happen was the sort of discovery / knowledge that Wnt signaling, which is a type of protein kinase signaling, it actually causes the stem cells to self-renew. So using these things called Wnt, stands for like wingless, which is a Drosophila gene carryover. It actually allows us to signal to the cells and tell them to stay alive and propagate. And without that, people weren't able to grow these stem cells before. So, and that's only sort of the exact cocktail of what media to put those, how to make it so that the cells are happy is recent and new. And a lot of it does fundamentally revolve around this specific signaling pathway called Wnt signaling. And because of our advanced or new knowledge of it, we're able to better grow the cells, and then it permits us to do these studies.
That's great. All right, I think we've got time for one more question. And so I'm gonna turn to the Q&A on this. And I think this is a question, how to switch the microenvironment that is inhibitory to cancer cells. So you're growing things in the dish, and it's really powerful to be able to isolate them. So how do you then translate it into an actual microenvironment?
Yeah, it's great. I mean, it's actually one thing that's very special and unique about the Koch Institute. So actually, my background is also as an engineer, as my PhD was also an engineer, and I used to be in Bob Langer's lab. And one of the technologies we're using in the Yilmaz lab is to nano encapsulate specific drugs or metabolites that we've identified that are useful for the stem cells, but potentially not good for the tumor cells. And that allows us to deliver those specific cytokines, drugs, or metabolites to the cells and create a new microenvironment that's selective. Even in the animal. And so it's a combination of these engineering technologies on how to do delivery, as well as the basic biological insights that allows us to tinker with how to, give an advantage to the normal cells compared to the tumor cells.
Great, that sounds so exciting. Thank you so much, George, and keep up the great work.
Thanks.
All right. So I want to just thank all of our presenters one more time. I'd also like to thank our judges. I didn't get to put this slide up on Tuesday, but I do want to today. This exhibition was the hard work of many, many people, including the judges who picked the images, and especially the researchers and the teams of microscopists, photographers, graduate students, postdocs, technical associates, even the principal investigators. So I want to thank everybody who participated in this, everybody who came and listened, and who shared their thoughts, questions. Like I said, the recording of this is going to be available online as soon as we get the transcription put together. And if you are interested in the next Koch Institute webinar on November 9th, we're going to be presenting Solutions with Insight, Pushing Boundaries, Breaking Barriers in Brain Cancer. Paula Hammond and Mike Yaffe and some of the folks from their lab and our clinical investigators are going to be telling you about how they're using multi-layered nanoparticles to break through the blood-brain barrier and attack glioblastoma with these synergistic combination therapies. So I hope you'll join us for that. If you're on our events mailing list, you'll get the notice probably in October. And so basically they're trying to deliver this 1-2 punch to brain cancer. And I always say when I start to get punchy, it's time to end the webinar. So I just want to say thanks again for coming. To our researchers, keep up the amazing work you're doing, and everyone else, please keep supporting the efforts to make the world safer, healthier, more beautiful. Have a wonderful, wonderful afternoon. And I'll leave these slides going just so everyone can see the images one last time and share any last thoughts that you have in the chat or Q&A.